Hi micro folks, this is Professor Carberry. It's Monday, June 8th, 2020. So I just discovered that two of the videos I made yesterday on the lactic acid fermentation of the milk and of the cabbage, I had my microphone muted. So we will be redoing those experiments or those videos today. So folks, this is the start of a new topic. It will probably be included in our topic prokaryotic ecology. And this is an introduction to the nitrogen cycle. So if we were meeting face to face, I would be writing up on the whiteboard this old cartoon of the nitrogen cycle, and we'll make sure, folks, that you have this as part of your lab packet. Okay. So the nitrogen cycle, folks, if I can see if I can get this up here correctly, the nitrogen cycle will start with molecular nitrogen that's in our atmosphere. So almost all of the air we breathe is molecular nitrogen, N2. Excuse me. And molecular nitrogen, folks, it's two nitrogen atoms covalently linked by a triple covalent bond. And triple covalent bonds are so hard to break. So that means that we organisms can't use molecular nitrogen as a source of nitrogen to make amino acids and proteins and nucleotides and nucleic acids. So we would say it's molecular nitrogen is not usable by us organisms. So what is so cool is that there's some bacteria that can take the molecular nitrogen, and our cat's meowing at me soon, guys. I hope that's not too distracting. So we have some cool bacteria that can take the molecular nitrogen and through an incredible enzyme called nitrogenase can break the triple covalent bonds and fix the nitrogen into ammonia, and it's fixed now in a usable form. We cellular organisms can use ammonia as a source of nitrogen to make amino acids, proteins, nucleotides, and nucleic acids, right? So this cool process, you guys, that some bacteria can carry out is called nitrogen fixation. And that's what we'll be focusing on a little bit later in the video, okay? And then, folks, there's another group of bacteria that can take the ammonia or ammonium and convert it into nitrites and then into nitrates. So this process is called nitrification. And again, folks, if we were like farmers, we would love this process because nitrification converts our ammonia into another usable form called nitrate, and plants love nitrates. Yeah. So if we were farmers or gardeners, we would love nitrogen fixation and we would love nitrification. Now, if we were farmers, we wouldn't like the last part of the cycle, which is called denitrification. And this is when denitrifying bacteria take the nitrate, the usable nitrate, and they convert it back into the unusable molecular nitrogen. So denitrification, right? So what we'll do, folks, in lecture and in future labs, we'll talk more about how denitrification can be beneficial or harmful depending on your point of view. But for this one, you guys, we just want to focus on the nitrogen fixation, the first step of the nitrogen cycle. And we just want to introduce you to a cool soil bacterium called Rhizobium. So Rhizobium, this is so fun, you guys. Rhizobium has co-evolved with a specific group of plants called legumes. And legumes, you guys, are beans, lentils, right? Peas, sweet peas, and peas, sugar peas that we would eat. They include peanuts, lupin, right? So this is a specific group of plants that have co-evolved to live with Rhizobium. So, and we're going to carry out this experiment, you guys. So when we plant our seeds, or excuse me, if we plant our beans, peas, clover, alfalfa, lentils in the ground, if the Rhizobium are present, the Rhizobium are going to invade the roots. And in response, the plant's going to form a little bump on its roots where the Rhizobium will live. And these little bumps are called root nodules. So you guys, today I was down in our community garden plot and I tried to plant lots of legumes because, again, the little Rhizobium that will live in the legume roots of these root nodules, they'll fix molecular nitrogen into usable ammonia. So this is a way for you to enrich your soil with usable nitrogen without having to use artificial fertilizers. And again, later on, you guys will talk about some of the problems with artificial fertilizers. So you guys, what I'm doing right now is I'm covering my keyboard because what I So sorry about that, folks. I have one headset to here and one headset is my microphone. So folks, you might not have heard any of that before. That's all right, I'll re-record it. But what we're doing right now is we are looking at the nitrogen cycle, okay? And we're looking specifically at nitrogen fixation, and we're looking at a soil bacterium called Rhizobium that has co-evolved with legumes. The rhizobium in the soil invade the legume roots. The legume forms this little bump called a root nodule in which the rhizobium lives. And it's in the root nodule the rhizobium carries out nitrogen fixation. So from our community garden, you guys, this morning, I was so excited. I like to grow legumes in the garden, and that decreases the amount of artificial fertilizer you have to use. So this is an old fava bean plant, and fava beans, you guys, let me see if I can see some here. Yeah, this one. So here are some young fava beans, okay, legumes, and I can hold on to these seed pods and dry them, and then use the fava beans inside to plant next year's crop, or if I want, I can eat them right now. They're really good. But what I was so excited about, you guys, and again, I sure hope you can see this. I've got to learn to be a better movie producer here. But what I can see, folks, is on the roots, there's these little bumps. Wish I could find a good one for you here. There's one right there, folks. Can you see that little bump? That little bump on the root right there? It almost looks like a little nose that's growing out of the root. That's a root nodule. And within that root nodule, the rhizobium are living. And what's so cool about the root nodules, they create an anaerobic environment. It turns out nitrogenase can't function unless it's in an anaerobic environment. So that's what the plant is doing to help the rhizobium out. So within the root nodules, the rhizobiums are just going nuts carrying out nitrogen fixation, taking molecular nitrogen, converting it into ammonia. And then the plant can use the ammonia to make amino acids, proteins, nucleotides, and nucleic acids. So the rhizobium will help the plant grow faster, stronger. If it's a food plant, it would be making more food for like animals or people to eat, right? And then, so if the rhizobium is helping the plant, right, we want to say, well, is the plant helping the rhizobium? And you guys, they are, because nitrogen fixation takes so much energy. It takes lots and lots of ATP, that energy currency of the cell. And so the rhizobium, they're going to need lots and lots of carbohydrates, lots and lots of sugars. So what the plant is doing up above the soil, right, where the sun is shining, the plant is carrying out oxygenic photosynthesis, taking inorganic carbon dioxide from the air and converting it into sugars. Isn't that just awesome, you guys? And then what the plant does is it can transport the sugars down into the roots. So the plant can feed the sugars to the rhizobium, so the rhizobium will have enough energy to perform the nitrogen fixation. And you guys, this, I wish I could get my head around here. I wish I had a giraffe head. There's all kinds of root nodules here. So folks, this is a type of symbiosis. A symbiosis is when two different organisms live together. This is a type of symbiosis that's the best kind of all. It's called mutualism, where both of the partners, the rhizobium and the plant, benefit. So the rhizobium is going to fix the nitrogen, so the plant will have ammonia, right, to make proteins and nucleic acids, so it's going to grow stronger, faster, produce more food. And then the little plant is carrying out oxygenic photosynthesis, so the rhizobium will have enough sugar to perform the nitrogen fixation. So this is so cool, you guys. I wish we could all live together in mutualism. All right. Now, another thing, folks, this was by accident, but when I pulled up the rhizobium, I actually pulled up one of its neighbors.